The transition beyond the fourth decade of life is not marked by a sudden biological collapse, but by a gradual recalibration of internal systems that have spent decades prioritizing growth, reproduction, and expansion. Around the age of 40, the human organism begins shifting its metabolic priorities toward preservation. This shift is subtle, largely invisible, yet measurable across nearly every physiological system. At the cellular level, aging reflects accumulated molecular stress interacting with declining repair efficiency. DNA within each cell experiences thousands of minor injuries daily caused by oxidative reactions, environmental exposures, and replication errors. During youth, repair enzymes rapidly correct these disruptions, maintaining genetic stability. Over time, however, repair fidelity decreases. Not because the body stops repairing itself, but because competing demands for energy and resources increase. Mitochondria, responsible for generating cellular energy, undergo structural and functional changes during midlife. These organelles rely on precise coordination between nuclear DNA and mitochondrial DNA. After decades of metabolic activity, mutations accumulate within mitochondrial genomes, reducing efficiency in ADP production. The consequence is paradoxical. Cells may receive sufficient nutrients yet produce less usable energy. Individuals experience this as fatigue, slower recovery, and reduced endurance. Simultaneously, the regulation of reactive oxygen species changes. While often portrayed purely as harmful, these molecules also function as signaling agents that regulate adaptation. With age, antioxidant defenses become less responsive, allowing oxidative damage to persist longer. Lipid membranes stiffen, proteins lose flexibility, and intracellular communication slows. One of the most significant changes after 40 involves insulin sensitivity. Muscle tissue gradually becomes less efficient at absorbing glucose from the bloodstream, particularly when physical activity declines. The pancreas compensates by producing more insulin, maintaining normal blood sugar levels temporarily. Over years, this compensation increases metabolic strain and promotes chronic inflammation, a condition sometimes described as metabolic aging. Hormonal signaling undergoes equally profound recalibration. Growth hormone secretion decreases, reducing the body's capacity for tissue regeneration. Testosterone and estrogen levels shift, influencing muscle mass, bone density, fat distribution, and mood regulation. These hormonal transitions are not failures, but adaptive responses to changing biological priorities. However, modern lifestyle conditions often amplify their negative effects. Muscle tissue begins a process known as sarcopenia, the gradual loss of muscle fibers and strength. Without regular resistance stimulation, the body interprets unused muscle as metabolically expensive tissue and reallocates resources elsewhere. Muscle loss further reduces metabolic rate, creating a feedback loop that encourages fat accumulation and insulin resistance. Adipose tissue itself changes character during midlife. Fat cells become more inflammatory, releasing signaling molecules that influence immune function and vascular health. Visceral fat, stored around internal organs, increases particularly when sleep quality declines or stress remains chronic. This type of fat behaves hormonally, altering metabolism throughout the body rather than simply storing energy. Cardiovascular structures also adapt with age. Arterial walls gradually lose elasticity as collagen cross-linking increases through glycation reactions, chemical bonds formed when excess glucose attaches to proteins. Blood vessels become less flexible, requiring greater pressure to circulate blood effectively. Over time, this contributes to rising blood pressure and increased workload on the heart. Meanwhile, the immune system transitions into a state scientists call immunosenescence. The production of naive immune cells declines, reducing the body's ability to respond to new pathogens. Existing immune cells remain active but often produce prolonged inflammatory responses. This imbalance explains why recovery from illness becomes slower despite continued immune activity. The brain undergoes quieter but equally important changes. Synaptic pruning continues, refining neural connections based on usage patterns. While certain cognitive processes may slow slightly, efficiency often improves through accumulated experience. However, chronic stress and poor sleep accelerate neural inflammation and reduce neuroplasticity, influencing memory and emotional regulation. Sleep architecture itself evolves after 40. Deep slow-wave sleep decreases, shortening the period during which growth hormone is released and cellular repair peaks. Circadian rhythms become more sensitive to light exposure, meaning irregular schedules exert stronger physiological consequences. Perhaps most importantly, cellular communication begins to lose precision. Hormonal signals, inflammatory markers, and metabolic cues become noisier, leading to reduced coordination between organs. Aging is not merely damage accumulation, but a loss of systemic synchronization. Despite these changes, the body remains highly adaptable. The biological processes shifting after 40 are responsive to environmental signals. Movement, nutrition timing, sleep consistency, and stress regulation directly influence how dramatically these transitions manifest. The organism is not entering decline by necessity. It is entering a phase where maintenance becomes more dependent on behavior than biology alone. After 40, the balance between anabolic and catabolic processes becomes increasingly sensitive to daily habits. During earlier decades, resilience masks physiological inefficiencies. The body compensates aggressively for poor sleep, excess calories, or chronic stress. Midlife reduces this margin for error. Biological systems begin reflecting accumulated patterns rather than temporary deviations. Metabolism demonstrates this shift most clearly. The flexibility to switch between carbohydrate and fat metabolism declines when sedentary behavior dominates. Cells become metabolically rigid, relying heavily on glucose while losing efficiency in fat oxidation. This rigidity contributes to energy instability, cravings, and gradual weight gain despite unchanged caloric intake. At the molecular level, nutrient-sensing pathways such as mTOR and AMPK regulate whether the body prioritizes growth or repair. Constant nutrient availability keeps growth pathways activated, suppressing cellular cleanup mechanisms like autophagy. Reduced autophagy allows damaged proteins and dysfunctional mitochondria to accumulate, impairing cellular performance. Periods of metabolic rest restore balance by activating maintenance programs that recycle cellular components. Inflammation becomes a central mediator of aging progression. Low-grade inflammation alters endothelial function, impairing blood vessel responsiveness and nutrient delivery. In the brain, inflammatory signaling interferes with neurotransmitter balance, influencing mood stability and cognitive clarity. These effects often appear gradually, perceived as normal aging rather than modifiable biological processes. Bone remodeling also shifts during this stage of life. Osteoblast activity, responsible for building bone, slows relative to osteoclast activity, which breaks bone down. Mechanical loading through resistance exercise stimulates bone preservation, illustrating how physical signals directly influence structural biology. The endocrine system continues adapting to environmental feedback. Cortisol rhythms, ideally highest in the morning and lowest at night, become disrupted by chronic stress and artificial light exposure. Elevated evening cortisol interferes with sleep onset and repair processes, creating a cycle of fatigue and metabolic imbalance. Mitochondrial renewal remains possible but requires stimulation. Exercise, caloric moderation, and temperature variation activate pathways, encouraging mitochondrial biogenesis, the creation of new energy-producing organelles. Without these signals, mitochondrial populations age collectively, reducing cellular vitality. Neurologically, the brain retains remarkable plasticity. Learning new skills, engaging socially, and maintaining physical activity promote the release of neurotrophic factors supporting neuron survival. Cognitive decline is therefore influenced as much by behavioral environment as chronological age. Gut microbiome composition also evolves after 40. Reduced dietary diversity and increased processed food intake decrease microbial richness, influencing immune regulation and metabolic signaling. Beneficial bacteria produce short-chain fatty acids that support intestinal integrity and reduce inflammation. Their decline contributes to systemic aging processes. Skin aging provides a visible reflection of internal biology. Collagen synthesis decreases while degradation increases due to oxidative stress and glycation. Reduced circulation limits nutrient delivery, slowing repair. Hydration balance changes as cellular turnover decreases. What becomes increasingly clear is that aging after 40 is governed by signaling patterns rather than irreversible deterioration. Cells interpret lifestyle inputs continuously, adjusting gene expression in response. Epigenetic markers shift depending on sleep, nutrition, movement, and stress exposure. Consistency becomes more powerful than intensity. Small daily behaviors repeated over years produce measurable biological outcomes. Regular physical challenge preserves muscle and metabolic flexibility. Stable sleep schedules restore hormonal coordination. Nutrient-dense diets support enzymatic reactions essential for repair. Psychological resilience also exerts measurable physiological effects. Purpose, social connection, and emotional regulation reduce chronic stress signaling, indirectly improving immune and cardiovascular health. Longevity research increasingly recognizes emotional environment as a biological variable influencing aging trajectories. The body beyond 40 is neither fragile nor fixed. It operates under different rules that reward alignment with biological rhythms and penalize chronic imbalance more rapidly than before. Repair systems remain active, but they require clearer signals to function optimally. Biological age begins diverging significantly between individuals during this period. Two people of identical chronological age may display dramatically different metabolic health, cognitive function, and physical capacity depending on accumulated lifestyle signaling. Aging continues, yet its pace reflects interaction between environment and cellular adaptation. The organism remains capable of renewal when conditions encourage repair rather than constant growth pressure. Inside every tissue, mechanisms designed for survival persist, waiting for signals that prioritize maintenance. The years after 40 therefore represent not simply decline, but a transition into a phase where conscious behavior becomes the dominant regulator of biological destiny.